Good morning, my name is Raylea and I'm from the Tasmanian Institute of Agriculture. I'm currently coordinating with State DPI surveillance of TPP throughout the eastern seaboard of Australia. The topics I'll present today will cover a brief TPP identification, field monitoring of the pest, farm biosecurity and state requirements for surveillance, how to be involved and where to report sightings of suspect insect or crop damage that may be affiliated with TPP. The tomato potato psyllid belongs to the order Hemiptera, or as they are commonly called, bugs. In this order you'll also find aphids, whitefly and scale insects, which I'm sure you're all very well familiar with. All insects from this order feed on plant tissue and excrete excess sugars or honeydew. Unfortunately, TPP is a vector for a bacterium called Liberiobacter candidatus solanacerium, or CLSO for short. The psyllid alone causes major damage to plants, but when associated with the bacteria, the damage is exacerbated. Psyllids become infected with the bacteria by feeding on infected plants. Interestingly, plants that are infected by the bacteria are more attractive to non-infected psyllids. The infected psyllid then continue to transmit the bacterium onto other plants, but they can also transfer the bacteria through to their offspring, which is one of the major factors that results in rapid geographical spread. Even though we do not have the bacteria in Australia, it is still essential to understand how this is spread so that quarantine measures can ensure the risk remains low and reduces the likelihood of its arrival. There are four main pathways of transmission. Pathway 1 is through the fruit, where the bacterium can be transmitted by psyllids feeding on fruit. However, in the absence of the psyllid, this cannot be transmitted through seed of tomato or from the fruit to a new host. Pathway 2 is through stored infected tubers, as these can become infected plants. However, viability of these plants is low. The plants are generally stunted and do not survive. If potatoes are infected early in the season, the plants generally die and do not produce any tubers. If the plant is infected with the bacterium during the mid-season, the tubers cease to develop. If the plant is infected late in the season and these tubers are harvested and stored, zebra chip development has been known to occur. In fact, 44% of infected tubers in storage have developed low viability plants when grown in glass houses. Pathway 3 is through nursery stock and grafting. Movement of infected nursery stock can spread the bacteria, and if infected plants are grafted onto non-infected plants, the bacterium can be passed through. Pathway 4 is through the psyllid. This is the pathway of greatest concern, as they can carry the bacterium, transmit it to host plants, are quite mobile, and can transfer the bacterium through to their offspring. It's thought that the transovarial transmission helps maintain the disease through overwintering psyllids, and also assists the spread of CLSO over large geographical ranges. The tomato potato psyllid is native to North America, with its distribution covering much of the central regions of USA, including Arizona, California, Colorado, Idaho, Kansas, Minnesota, Montana, Texas and New Mexico, and is present as far south as Guatemala and Honduras. The psyllid is also found in New Zealand and Western Australia. Generally, the bacterium CLSO shares its geographic distribution and can be found in all other regions where the psyllid is present, but currently no detection of the bacteria has been found in Australia. Why, you might ask? Well, this seems to be just down to luck. The psyllids are termed hot versus cold, where hot psyllids carry the bacterium and cold psyllids do not. So far it looks as though the psyllids that entered Australia were cold and did not have the bacterium. There has been extensive testing of psyllids and plant material from Western Australia to determine this, and so far nothing has been found. Current research is underway to ascertain how the psyllid arrived in Western Australia to mitigate the risk of getting a hot psyllid, and these include wind models, human traffic, and movement of plant material. The tomato potato psyllid is polyphagous, which means it can feed on a range of host plants exceeding 20 plant families. When it comes to completing development, there are more than 40 host plant species which all occur in the family Solanaceae. These include tomatoes, potatoes, eggplants, capsicums, chilies, tamarillos, and some Convolvulaceae sweet potato families. To date, it is not known if the bacterium has the same detrimental effects on the tubers of sweet potatoes as it does on normal potatoes. However, there are several non-target plants that adult psyllids are capable of surviving long periods of time on. These include African boxthorn, poroporo, thorn apple, apple of Peru, Jerusalem cherry, field binweed, Chinese boxthorn, just to name a few. In fact, in New Zealand, all life stages of TPP were found on these plants all year round. Furthermore, a study in the US showed that adult psyllids could survive between 17 and 96 days on non-target plants such as the matrimony vine, which is thought to be where the resident populations of adults reside over winter. TPP numbers vary from one year to the next because the life stages are severely impacted by extreme hot or cold temperatures. Temperatures above 38 degrees for prolonged periods of time result in nymph and egg death and adults cease laying. However, tomato potato psyllid adults seem to be reasonably cold tolerant, surviving in regions that experience frost and snow, and some even surviving temporarily at -15 degrees Celsius. When monitoring your crops for TPP, you'll be looking for any of the three stages of development: egg, nymph, and adult. A single female psyllid can lay between 300 and 500 eggs, averaging 350, with some records of females laying up to 700 eggs. The eggs you can see here in the red circle take approximately 3 to 9 days to hatch. The eggs are extremely small and will be very tricky to spot. The best locations to look will be on leaf margins, petioles and stems, or on stalks. The eggs are found on little stalks and appear yellow or orange in colour. However, eggs being so tiny can be confused with other eggs commonly associated with crops. So the leftmost image is a TPP egg, the one next to it on the longer stalks are lacewing eggs, and the ones below are whitefly. The second stage are the nymphs or juveniles. Nymph stages generally take 12 to 44 days from first instar through to adult. Nymphs of TPP are also quite tiny, measuring between 1 and 2 millimeters in length. Newly hatched nymphs are lime green in colour, progressing to a yellow-green with later instars. All stages of nymphs will move slightly when prodded, unlike other nymphs. In this image here, you can see newly emerged nymphs through to fourth instar. They have the characteristic red eye spots. This image here shows the fifth and final instar of the TPP nymphs. Key features to look for will be the wing pads and the waxy filaments that go along the lateral margins of the body. The two other images here show you what you would typically see underneath a leaf with an infestation of TPP. You'll see many different stages. In the top image there you can see two orange-coloured adults. Adults, when they newly emerge, are orange in colour and take a few days for the pigmentation to go to the typical TPP colouring, such as the black with the white bands. In the bottom image here you can see some sugary residue excreted by the nymphs. This is an attraction for ants as well. The final stage are the adults. A common name for the adults is jumping plant lice, as they're renowned for making rapid jumping actions when disturbed. This is generally a straight up and down action from the plant. Adults generally survive between 16 and 97 days and can have between 3 and 4 generations a season. Again, this is temperature dependent and could be greater given the right conditions. Adult TPPs are also 2 millimeters in length. The image here shows some newly emerged psyllids and some fully pigmented psyllids with the clear casing of the final instar nymph where the adult has hatched. Adults, females, can mate 3 to 4 days after hatch and can mate several times during their life cycle. Here is a large illustrated image of TPP. Some of the key features that will be recognisable for TPP adults are the erect transparent wings and the prominent white band that goes across the abdomen. And there's a reference there of a 5-cent piece which the represents the size of the psyllid next to it there. There's a significant edge effect with TPP infestations, with primary infestations occurring at the crop edge and approximately 5 metres inwards. There is a preference for the underneath side of leaves, so when you're monitoring your crops, randomly choose around 50 leaves to turn over and inspect. You'll likely see all stages of development. Also take note of any insects jumping directly up in front of you. It is possible to hold an open Ziploc bag over the plant to catch the psyllid should you wish to have one identified. Some signs to look for that may indicate an infestation of TPP and CLSO damage. For psyllids, the damage includes psyllid yellows, where you get chlorosis of the foliage. It's believed that this is caused by a toxin excreted by the psyllid during feeding. Other symptoms include stunted growth, necrosis, and in large infestations, plant death. Symptoms can be seen 4 to 6 days after injection of the psyllid toxin, and in a heavy infestation, usually 2 to 3 weeks. Symptoms to look for with CLSO infection include the typical zebra chip appearance of potato tubers, where the tuber has collapsed stolons, browning of the medullary ray, vascular ring, and necrosis, which are all amplified upon frying. Foliar symptoms of CLSO infection resemble psyllid yellows and purple top, and include upward rolling of the leaves, apical tapering of the leaves giving a spiky appearance, stunted growth, discoloration, shortening internodes and swollen nodes, leaf chlorosis and scorching, and in the case of tomatoes you get fruit abortion, or multiple misshapen fruit and rapid plant death. Here are some pictures to demonstrate these symptoms. You have necrosis of the tips of the leaves, stunted growth and chlorosis, some aerial tubers, leaf purpling, and a proliferation of tubers. Also, the classic symptoms of zebra chip from these pictures here are fried potato. These images here show some tomato damage. You've got leaf purpling, leaf cupping or rolling, which is also signs of European corn borer damage, but in this case the leaves are not brittle. You have leaf chlorosis and some misshapen fruit. As you're probably all aware, identification of TPP and crop damage is not straightforward. Many of the foliar symptoms resemble many other diseases known to these crops. There are also over 400 native psyllids, but many of which are associated with eucalypts or wattles. But they also secrete a honeydew. For this reason, it's important to familiarise yourself with what TPP looks like and regularly monitor your crops. To ensure your risk of an incursion is low, an important aspect is improving your current farm biosecurity practices. Callum Fletcher at AusVeg is coordinating workshops and providing resources around farm biosecurity. This is being done through focus groups and support networks that can collaboratively work together in similar regions to assess benefits of certain farm biosecurity practices, discuss new initiatives, and improve not only their biosecurity but that of their region. Some of the steps include a risk assessment that will essentially provide you with the tools to understand your risk and the regions of farm biosecurity that need to be prioritised. Discover the gaps in your practice which could be improved, and develop a risk management plan to integrate these changes to become routine practice. This is an example of a risk matrix that can be used to assess your risk of certain plants and pests and diseases. Some of these things to consider are the financial implications, the legal obligations such as reporting, the reliability of your current farm biosecurity practices, whether they ensure your risk is low, if there will be a reduction in marketability of the product, and what would be the associated loss of revenue be for the season, and how long would these impacts last. Some of the things to consider are the pathways onto your farm. Vehicles and equipment, where do they come from? Have they been used on other farms? What procedures can you improve to minimise transfer of pests and diseases through farm vehicles, such as including washdown stations or restricted movement around the farms? Packaging, bins and pallets. Do they get recycled on your property? What's the risk of infected soil or plant material being transferred to new crops? Is there a cleaning procedure after each movement, and where do they come from and where do they go? What signage do you have around to inform visitors and staff of the procedures in place? Who do you have regularly entering the property? Volunteers, contractors, tourists, news reporters, service providers, family members and school groups? What procedures do you have in place to check clothing, footwear and tools for organic matter and soil? Should you put in place cleaning facilities including foot baths and brushes that are accessible for visitors and staff? Do you have an induction for all staff and visitors, raising awareness of biosecurity expectations prior to moving around your farm? Do all visitors report to management and sign in a register? Wastes and weeds. Plant waste and weeds can be an effective incubators of plant pests and diseases. Cucumber green mottle mosaic virus and potato spindle tuber viroid are examples of pests that can hide in organic wastes or surrounding weeds when primary hosts are not available. So it's essential to store waste away from growing areas and water sources, dispose of waste as soon as possible, which can be done through burial, burning or composting, controlling weeds and maintaining a weed-free buffer zone around growing areas to prevent incubation and transmission of plant pests back into the crop. Planting and materials. Fertilisers, compost, plastic mulches, seedlings, seeds and seed potatoes, as well as other planting material, can incubate plant pests and disease. Do you know the source of planting material, and is this from a reputable supplier? Do they treat for pests as required? Follow seed testing requirements for imported seeds and arrange testing of seeds if necessary to keep any tests for plant pests on record. Record keeping is an important part of farm biosecurity too. Having a good system in place, especially records of monitoring efforts, even if sightings of TPP are nil, as this information can be used for declarations of area freedom later on. Currently, it's a state requirement to provide area freedom certificates to prove you are free from TPP. These certificates maintain your market access. This can only be achieved by providing evidence-based data that the region is free from TPP. The surveillance programs aim to offer broad area coverage of host plants, growers and nurseries, and regions of high risk such as transport depots and community gardens. This data goes towards preserving existing trade between other states, and it also supports access to international markets. It complements your monitoring efforts by providing entomological certainty that you are free from TPP. How is this being coordinated? The program we're running through TIA will assist other state governments with surveillance. We communicate our surveillance efforts with each state DPI to ensure the best possible coverage and frequency of surveillance throughout the growing season. Our data goes towards area freedom for each state, and our records comply with national minimum data standards. We provide you with traps which are mailed out upon request. The trap packs come with sticky traps, plastic covers for completed traps, Ziploc bags to protect and package the traps into for mailing, and reply paid envelopes. We also have a team of entomologists assessing these traps. How to be involved in the project is quite easy. Simply contact us with the number of fields you wish to survey, a rough size of each field, and an address that we can mail the packs to. All packs come with instructions and guides for installing these traps. The traps are placed at crop canopy for around 7 to 10 days, and then they are collected. There is a sticky label on the Ziploc bags to record when the traps went out and got collected back in, what the GPS coordinates are of the field, and a property name, which could be as simple as the street name or the company name where the traps were placed out. Then you place the traps in the mail and they get delivered to us for assessment. Overall, the key message is that knowledge is fundamental for a response to pest incursions. In the case of TPP, the sooner we are aware that there has been an incursion, the quicker we can respond to contain and, best case scenario, locally eradicate the pest. If in doubt about a potential pest or plant damage on your property, please call the Emergency Plant Pest Hotline on 1800 084 881 or your local biosecurity number.